Hello students, welcome to EPG Pathshala. I am Dr. Renu Tyagi from Department of Anthropology, University of Delhi. Today I am going to speak on the module Evolutionary Primate Genetics from the paper Human Origin and Evolution. The learning objectives of the module are to describe the primates and their classification and to understand the characteristics of the primate. The another objective is to describe the ABO blood group system of primates, to know about the chromosome number change among the primates, and to understand the impact of evolution on evolutionary primate genetics. Now let's begin with the introduction. Primates are the mammals that separated from a primitive group of the mammals, perhaps the small primitive insect-eating mammals, the insectivorous, some 60 million years ago. Mammals are one of the 5 taxa of the vertebral animals. The vertebrates are the animals with the vertebral column. This including not only the mammals but the fish, amphibians, reptiles and the birds. Mammals are characterized as the warm-blooded vertebrates having an advanced reproductive system, mammary glands and fur or hair somewhere on their body. The first major definition of the primate was published by the British anatomist St. George Mivart in 1873. He wrote that primate as an order are ungulate, claviculate, placental mammals with orbit encircled by the bones. 3 kinds of teeth, at least at one time of their life, brain always with a posterior lobe and a calcarine fissures. The innermost digit of at least one pair of the extremities opposable, helix with a flat nails or none, a well-developed cacune, penis, pendulous testes, scrotal, always 2 pectoral mammae. The current definition of the primate is as follows. Primate have retained a generalized skeleton with 5 fingers and toes or pentadactyly. The innermost digit on the hand, the thumb and the foot, the big toe usually has enhanced mobility and there is a tendency for nail to replace the claws on at least some of the digit. There are also touch pads possessing the fingers and toe prints at the ends of the digit. The orbits are enriched with the bones, that is, there is a post-orbital bar present so that the eye is completely surrounded by a bone. A characteristic of all the primates is that the middle ear chamber or auditory bulla is formed from the petrous portion of the temporal bone. There is a retention of the clavicle in all primates which enhances the mobility of the shoulder joint. Now let's have a look at the characteristics of the primate. The number 1 is that the primate bear the flat nails upon their digits. This makes the grasping function of the hand and the feet easier. Number 2 is the presence of the well-developed clavicle is a characteristic feature of the primates. The orbits are completely surrounded by the bony rims. Their limbs are prehensile and adapted for the arboreal life. This was probably an earliest mammalian characteristic. Either the thumb or the great toe or both are opposable. This possibility, which is an ancient mammalian feature, has been retained by the primates during their long arboreal life. The teeth are adapted for mixed food vegetarian and animals. 3 kinds of teeth are present in the primate at least at one time of its life. The teeth are differentiated into incisor, canine and molar to serve different functions of cutting, holding and grinding, etc. In the primates, the mammae are 2 in number and they are pectoral in position. The nursing habit of the mother determine the position of the mammary glands. The development of the brain of the primates shows wide variation, but a calcarine fissure and a posterior lobe is always present in the primate brain. The primate possess pendulous penis. The character is shown by some other mammals also. They have a well-developed cecum, that is a sac connected with the intestine of an animal. The stomach is simple, the femur has never third trochanter. The testes are descended into the scrotum. Of course, it is present in many other agile and active mammals also. Now let's have the classification of the order primates. The primates are broadly classified as prosimians and anthropoidea. The anthropoidea is further classified as the platyrrhinae, that is New World monkey, and catarrhinae, that is Old World monkey. The catarrhines are further subdivided as the Cercopithecoidea and Hominoidea. The Hominoideas are further classified as Hylobatidae, which includes the gibbon, Pongidae, which includes chimpanzees, orangutan, gorilla, and Hominidae, which includes the humans. Now let's have a look at the evolutionary primate genetics. First we'll talk about the blood group. Landsteiner discovery in 1901 of the blood group in humans initiated what turned out to be one of the most active and important research area in the clinical medicine of human genetics, and today organ transplant. As early as 1925, it was known that the red blood cells of the non-human primates had antigen, a substance that stimulates the production of antibodies similar to those of the humans. Then in 1940, Landsteiner and Wiener discovered the Rh factor in rhesus monkeys. One of the most important discoveries in the history of the blood group studies in primate. This discovery of the monkey type antigen in humans' blood was a great impetus to study the blood of the other non-human primates using other human antigens. Later, antisera were made from the red blood cells of the non-human primates themselves and then used to ascertain the specificities of the blood of other non-human primates. The former are known as the human type blood group, while the later are simian type blood groups. Information regarding the blood group of the non-human primates is much more limited than it is for the humans. At present, there are several separate blood group systems under study in non-human primates, such as the well-known ABO, MN, RH, Lewis and Duffy. There are blood groups data for all the anthropoid apes. Some species of Old World monkeys, very few species of New World monkeys and only a few prosimians. The ABO blood group is the well documented in non-human primates. Now let's talk about the ABO blood group system. The ABO group was the first blood group system discovered in the humans by Landsteiner in 1901. It has been classified and found in all primates that have been examined. Primates have 4 blood group type A, B, O and AB. Table 1 shows the 4 ABO blood groups of the humans and non-human primates. Table 2 shows the reaction on the ABO blood group. There are 2 agglutination antigen on the red cells A and B and 2 anti-A and anti-B substance present in the sera, which determines an animal blood type. Thus, red cell of a group A animal contain A but not B agglutinogen, while the serum has anti-B but not an anti-A agglutinin. If a transfusion is necessary, it should be from an individual whose blood cells do not become agglutinated when mixed with the blood of the recipient. In humans, persons with the blood group O are considered universal donor, while persons with the AB blood group type are considered as the universal recipients. These substances are present not only in primate blood but in their saliva as well. Since the ability to secrete the A, B and O antigen in the saliva is inherited as a dominant character. Also, since A and B substances are not normally detectable on the red cell of the primates, other than in the hominoids, saliva is used for identifying the ABO blood group in other primates. The ABO blood group frequencies for the anthropoid apes are presented as table 3. The 2 specimens of the gorilla and the simang have only B, while the pygmy chimpanzee has only the A group. These 3 taxa are monomorphic, that is one form with respect to the ABO system, while the other apes are polymorphic with the many forms. It is interesting to note that the common chimpanzee is the only great ape having the O blood group. Besides the obvious importance of the blood group in clinical situation, such as blood transfusion and cases of the materno-fetal incompatibility, they also have been of value in estimating the time of evolutionary divergence as well as in taxonomic classification and as genetic marker. For example, the fact that the ABO antigen are present in all primate studies to date would suggest that ABO polymorphism was present in the primate lineage prior to the separation of the old and new world monkeys. Another important contribution has been in the study of the hybridization of monkey population living in the overlapping geographical areas. This table 3 shows the ABO blood group of the lesser and the great apes. Now let's come to the RH blood group system. The RH system is of interest to the primate biologist because of its importance in evolutionary genetics and its practical implication for understanding the RH factor on human red cells. RH factor is originally found in the rhesus monkey, thus the RH symbol refers to the rhesus monkey. In humans, there is a condition known as the hemolytic disease of the newborn, that is erythroblastosis fetalis, which cause the oxygen-carrying red blood cells of the fetus to be destroyed. The result is usually death of the fetus. This rather rare situation, about 85% of the humans are Rh positive while 15% are Rh negative, occurs when the parents have incompatible Rh factors. For example, if the mother is Rh negative and the father is Rh positive, then the fetus is Rh positive. The hemolytic disease is due to the reaction of the antibodies stimulated within an Rh negative mother by the Rh positive cells of the fetus. In subsequent pregnancies, these anti-Rh positive antibodies from the mother enter the fetal circulation and destroy the fetal cells, resulting in a severe anemia. It is very interesting to note that among the anthropoid apes, only the red cells of the orangutan fails to react the homologous with the human Rh factor. Now let's see what are the chromosomes. Chromosomes are the colored bodies present in each cell of an organism and carry the genetic information or the DNA, which is arranged in each chromosome as genes. The study of the chromosome, known as the cytogenetics, has become the important branch of the molecular biology. Chromosomes appears as the paired structure consisting of two identical chromatids joined to each other at the primary constriction by the centromere and have different morphological characteristics that can be used in their identification. A chromosome is metameric if the centromere is near the middle of the chromatid. If the centromere is near the end of the chromatid, it's the acrocentric. And if the centromere is at the end of the chromatid, it is telocentric. When the centromere is between the middle and terminal position, it is subterminal. Chromosomes are counted and studied during the cell division, especially during the metaphase, for it is during this time that the chromosomes separate and become most prominent as they line up along the plane of cell division. The number and type of chromosomes in an organism cell constitutes its karyotype. A specific karyotype is characteristic for a species. For example, humans have 46 chromosomes known as the diploid or 2n number. For example, humans have 46 chromosomes known as the diploid or 2n number, consisting of 23 pairs of chromosome, of which 22 pairs are autosomes and 1 pair are sex cells or sex chromosomes, which is XX in female or XY in male. The number of the chromosome present in the human germ cell is 1, that is half of the diploid number, or 23, and is known as the haploid. Karyotype vary a great deal among the living primates. These 2n range from 20 to 80, which indicates much selection during the course of primate evolutionary history. Changes in the number or shape of a chromosome results in rearrangement of the genetic material, which is often fatal or deleterious to the organism. Chromosomal rearrangements include the loss of the chromosome material, that is deletion, or duplication of the material, turning the pieces of the chromosome upside down, that is inversion, and translocation, which is exchange of the material between the chromosomes. Since such changes are rare and a given number and form of chromosomes remain stable for a particular species, there is probably strong selection pressure against such events. However, these events do occur in population, and if they take place over many generations, the result is chromosomal evolution. In other words, these rearrangements constitute the raw material for the karyotype evolution, which may result in different species. The number of chromosomes, that is 2n, varies among primates. For example, some species of the lepil lemur have the lowest number of the chromosome recorded among primates, where 2n is equal to 20, while the specialized little tarsiers have the most, that is 2n is equal to 80. Among the New World monkey, chromosome number range from 20 to 62. The 2n chromosome number of the Old World monkey varies from 42 to 72. The macaque and the baboon all have a 2n of 42. The chromosome number range from 44 to 52 in hominids. The highly derived chromosome of a gibbon, that is 2n is equal to 38 to 52, have undergone rapid and extensive changes since separating from the other hominids. Gibbon have mostly metameric and submetameric chromosome, although the Symphalangushas a single pair of the achromatic chromosome. The great ape have 2n is equal to 48 chromosome and all have 3 types of chromosome. The human, that is 2n is equal to 46, has resulted from the fusion of the 2 acrocentric chromosome to form the number 2 chromosome of the humans. Humans have all 3 types of the chromosome. The sex chromosome are similar, X and Y in the great ape and humans, that is X chromosome is submetacentric and the Y chromosome is acrocentric. The 2n number of the primate chromosome number is represented by the prosimians, which is 20 to 80, while the great ape and humans have the smallest range of the chromosome, which is 46 to 48. Different species may have the same number of chromosome, but their form may be quite different. So, the characterization of a species karyotype must consider both the number and the form of the chromosome. Now, let's talk about the deoxyribose nucleic acid, that is DNA. One of the most important biological discoveries of the 20th century was the identification of the structure and the function of the deoxyribose nucleic acid, that is DNA. This material, which with protein makes up the majority of the cell nucleus, contains the genetic message that enables the cell to replace itself, as well as providing the information necessary for the cell's many other functions. The essential process of the life, therefore, is replication, and it is important that the coping process proceeds as accurately as possible. However, mistake, that is mutations, do occur in the coping process, but they are usually harmful to the organism. Occasionally, a mutation happens to be beneficial to the organisms and selection occurs and the mutation survives and spread through the population. The result is evolution by natural selection, which through millions of years has produced the diversity of the life present today on Earth. Advances in the DNA technology have made it possible to sequence the gene and compare them with the genes from the other species. Also, fragments of the DNA from 2 different species can be combined into a hybrid segment, a process called DNA hybridization. There are 4 types of bases in DNA: adenine, guanine, cytosine and thymine. The number of the matches and mismatches between the bases on the hybrid strands of each DNA fragment allows the determination of the degree of similarity between the 2 species at the DNA level. Such studies have provided information on the degree of genetic relationship among primates and have demonstrated that the DNA of the chimpanzees, gorilla and humans differs only about 2%. This means that when strands of DNA from any two of these animals are combined, about 98% of the bases match. Humans differ from the orangutan by about 4% and from baboon by about 8%. This similarity of the DNA sequence among primates taxa indicate their degree of biological evolutionary relationship. Now let's summarize the module. Primates are the mammals that separated from a primate group of the mammal, perhaps the small primitive insect-eating mammals, the insectivorous, some 60 million years ago. Lensteiner's discovery in 1901 of the blood group in humans initiated what turned out to be one of the most active and important research area in clinical medicine of human genetics. The study of human genetics through blood group, chromosomes, DNA and their similarity with the other primates has led the evolutionary history more predictable. ABO blood group system has obvious importance in the clinical situation such as blood transfusion and cases of materno-fetal incompatibility. They also have been of value in estimating the time of evolutionary divergence as well as in taxonomic classification and as the genetic markers. For example, the fact that the ABO antigens are present in all primates studies to date would suggest that ABO polymorphism was present in the primate lineage prior to the separation of the old and the new world monkeys. Different species may have the same number of the chromosomes, but their form may be quite different, so that the characterization of a species karyotype must consider both the number and the form of the chromosome. One of the most important biological discoveries of the 20th century was the identification of the structure and function of deoxyribose nucleic acid, that is DNA. The essential process of the life is replication, that is the copying process. Any mistake or mutation in copying process results in the change in the genetic makeup of the organisms. Sometimes these mutations are beneficial to the organisms and selection occurs and the mutation survives and spread through the population. The result is evolution by natural selection, which through millions of years has produced the diversity of the life present today on the earth. Thank you.